So I'm Jonathan Pritchard, and I'm a professor of biology and genetics at Stanford University.
Can you describe your graduate school experience in Marcus Feldman's group?
So Mark's really had a unique place in sort of establishing mathematical population genetics in sort of the human genetics world. So he so both on the research side as well as in terms of training an enormous number of people. So on the research side, he's he kind of developed a lot of mathematical theory, for example, in understanding cultural evolution, as well as his work with Luca Cavalli-Sforza in particular on developing the Human Genome Diversity Panel and doing a number of really key pioneering studies in understanding patterns of human genetic variation. So when I came to Stanford as a graduate student in 1994, they were in the process of putting together the Human Genome Diversity Panel at that time, and the lab tools for surveying larger numbers of markers were just coming online at that time. And so I was interested in understanding or in using genetic variation to study human history. And so Mark's work and sort of establishing all of those tools and the mathematical theory was central to what I was doing, as well as a lot of people in the field. David Goldstein and Magnus Nordborg graduated just as I was leaving, and then I worked closely with Carl Bergstrom, who's at the University of Washington now, Michael Lachman, David Ardell, Lauren Ansel Myers. So we had a very interactive group of graduate students. And then in my last year, Noah Rosenberg joined the lab, and actually worked very closely with him in my last year. Noah wanted to do a rotation with Mark, but Mark was going out of town, maybe even a sabbatical. And in my last summer, which was basically his first summer in the lab. And so we wound up working very closely together on a project that, in those last few months, which sort of laid the groundwork for a lot of the work that we did together over the next few years. I considered going to University of Chicago very seriously at that time. So I would say the main difference between them and Stanford was that University of Chicago kind of had a stronger evolutionary population genetics with in Drosophila kind of flavor. And so there were really great people there, and I almost went there, but at Stanford there was a heavier focus on human population genetics through Mark and Luca, and then others who I didn't actually know at the time that I was making my decision, like Neil Risch and David Cox, and so those were some of the things that drew me to Stanford at that time. So we actually had an enormous amount of freedom there, and I would, we'd often work for a couple of months without talking with Mark necessarily. Like he had his own projects. He traveled a lot, but when we wanted time with him, he was very generous. And I think as a lot of his mentoring role was sort of really high level and helping us to develop our careers. And he's been extremely supportive of me over the years, but at the same time giving us huge amounts of freedom to kind of explore our own ideas.
What contributions did the Human Genome Diversity Project make?
So I think the Human Genome Diversity Panel made a number of major contributions. So one was this idea of collecting samples of individuals who would represent who would represent different populations, and from which we could learn about sort of global patterns of diversity. And that kind of structure has was in a sense repeated by the HapMap project and the 1,000 Genomes project and other sampling projects since then. So I think that the concept of that kind of sampling was really important. A second major contribution, I think it's fair to say, is the work that Noah Rosenberg and Mark Feldman and I and others did on understanding human population structure using the HGDP. A third kind of area has been work on sort of specific patterns of variation. So understanding the structure of linkage disequilibrium and how that relates across different populations, or looking at how genetic diversity declines with distance from Africa. So that was a first done in a paper by Sohini Ramachandran around 2005, which was a really important result that was only possible because they had gone to this enormous amount of work of creating this panel of individuals.
How did Neil Risch influence your work?
So Neil was a professor in the genetics department when I was a graduate student. Now, I was in the biology department at that time as a PhD student, but I started taking classes in the genetics department whenever something came up that looked interesting to me. And so Neil taught a series of classes there. The first one was with David Cox, who was another professor in genetics, and that was really my first introduction to human genetics in sort of a disease, from a disease point of view, kind of going beyond human history kinds of questions, which I had read about a bit up prior to that. And so then after I took that class, I thought, this is really interesting. So I took Neil's next classes, which were on statistical genetics. And so he had this pair of classes where he talked about sort of mathematical and statistical techniques in human genetics. So for example, techniques for computing likelihoods and pedigrees and things like this. Through that, I really became interested in in kind of the genotype phenotype question. So basically in understanding how genetic variation causes disease, and I started to think about how population genetics tools that I knew a lot about from my PhD work could be used in understanding sort of more mainstream human genetics questions. And so one paper that Neil had written that was influential on the whole field as well as on myself in particular was the Risch and Mirakangas paper from around 1995 or 1996 that argued that association mapping was going to be much more powerful than linkage mapping. And it's a very elegant paper. It's 2 pages, and I still sometimes assign it to my students when I teach. It's just a very very concise paper that I think really changed the direction of a field. So anyway, all of those things got me interested in human genetics through Neil's classes. But there was one lecture in particular that had a big impact on me, which was when Neil was teaching the transmission disequilibrium test, the TDT test, and so he made this comment that we don't really know how to deal with population structure for case control studies of association mapping, and instead the standard in the field at that time was to use these family-based studies. And so through my interactions with Luca Cavalli's lab, I knew about the work that they had been doing on using genetic markers to cluster individuals from different populations. And so they this is work from Anne Bocock and Joanna Mountain and others. And so they had shown that using microsatellites that you could basically distinguish people who came from different continents. And so I thought, well, maybe we could put that idea together with the case control study question and actually use genetic data to solve this problem. And that would potentially have some big advantages, because then you could do case control studies without requiring family members, and so that would really simplify studies very much. And so I went to that one lecture, and it actually changed the course of my research for the next half dozen years or so.
How did you become focused on how to infer population structure from different kinds of data?
I think that was probably about April of the year that I graduated, and so Noah and I went back to our office and started talking about this, and so then we started thinking, well, let's suppose you've got a case control study And let's suppose that back then people were thinking in terms of sort of candidate studies of association. So you'd have some genes that you're particularly interested in because you thought they might be involved. And of course, that turned out that paradigm didn't really work very well, but in retrospect. But at the time, what I thought was, well, maybe we could do genotyping or sequencing of these candidate genes, but also add on a set of additional unlinked markers elsewhere in the genome that would be basically a control set. And so then the first paper that Noah and I wrote discussed statistics of this. So if you have a control set of SNPs or other kinds of markers, and how many would you need to convince yourself that there wasn't strong structuring between cases and controls, and that the signals you got at the candidate genes were reliable.
How did you come to work with Peter Donnelly at Oxford?
Mark's approach to population genetics I would characterize as being very mathematical. And by the end of my PhD, I decided that I wanted to become better at statistics, to add a statistical component to my work. And so Peter Donnelly had published a paper around 1996 that kind of exemplified the sort of work that I wanted to do. And so I contacted him, and one thing led to another, and so I wound up going over to Oxford to study with Peter. But it was really because I thought that he sort of had a statistical perspective on population genetics that I could learn from.
How did the structure algorithm for studying population structure come about?
Out of all the projects I've done, that's the one that probably unfolded the fastest. So when I first went to England, there was this several months program that was going on at the Isaac Newton Institute in Cambridge, and so I went there, and Peter was spending the autumn quarter there, and so was Matthew Stevens, who was another postdoc in the Donnelly lab, who had been there for a year when I arrived, and so maybe a week or two went by before I got an extended meeting with Peter, and he said, well, what do you want to work on? And so I told him at that time about the problem that I'd been working on with Noah, and I said, well, here's how far we've gotten. We think we know how to detect population structure when it's contaminating a case control study, but we don't really know how to correct for it. And we'd been doing a bunch of work on clustering methods, but we didn't really have anything that we were happy with. And so Peter liked this question, I think, and he at that point suggested that we could use Bayesian clustering methods. And so he was at that time, I think, influenced by a paper that had come from the DNA forensics literature. So Catherine Ruder and Bernie Devlin and others have done some work on this. And so he kind of suggested this framework, and Matthew Stevens who became a very long time collaborator and friend, was with us, and so we kind of went to the board straight away and started brainstorming models. And Matthew knew a lot more Bayesian statistics than I did, and so so Peter and Matthew started working out ideas, and then I was kind of making suggestions to the extent that I could, following along. And so by the end of about 2 hours, we had actually sketched out the whole model, and I think I have this in my notebook somewhere. And so it was the model that was sketched out on the board essentially wound up being the final structure model. And so then I went back to my office and programmed that, and it took a few days to program it, and I had some data that Lynn Geordie had given me kindly, and back then there wasn't a lot of human variation data that was suitable. So, but anyway, I took this data set and I ran it, and I'm sure there's a little bit of debugging, but within a week or 10 days or something, I found that we could actually get perfect clustering of the individuals from different continental groups from that, and so that example's in the initial paper. And so of course, having done that work, which took about 10 days, of course, then, it's a long road from that to actually having a paper that you submit. But, from that point, it was immediately clear that we had something that was interesting and that worked really well.
Why is population structure important for understanding human genetics?
So population structure refers to the idea that populations that are to some extent distinct tend to have different allele frequencies. And so, all human populations descend from, a single out of Africa event something like 80,000 years ago. But since then, there's been limited migration between different continental groups. And then when you look within continents, populations have been separated over shorter periods of time. And that partial separation means that allele frequencies can drift to slightly different frequencies in different populations. So when you look at individual SNPs, you don't see hard differences between populations. Generally, essentially every allele will be present in all populations, aside from very rare alleles. But the idea with a method like structure is that we can use small allele frequency differences to identify the ancestry of individuals.
What makes the structure algorithm successful?
There's been there's been a lot of work on on models that are trying to, or statistical methods that are trying to infer allele frequency changes over time. And if you measure allele frequencies in different populations, then you can, to some extent, you can infer what the past allele frequencies may have been. Of course, more recently with the advent of ancient DNA, that's helped with that significantly. I think actually one of the reasons why the structure method was so successful was that it actually ignored a lot of those complications. And so in some sense, it's not really a full population genetics model. It's more of a clustering model that allows us to identify groups of individuals who have similar alleles. And then it doesn't really talk very much about sort of the historical process that led to that. But by ignoring the historical process, it kind of allows us to get a lot more power out of identifying individuals who look similar in genetic terms. And so I think it kind of raised a new set of things that people could look at. The first motivation that we had was the idea that we would use this for association mapping. And so we had a follow-on paper where we talked about how to do this. But essentially the idea was that if you have case and control individuals, then when there is population structure, that can cause confounding if you don't know about it. But our idea was that we would run the structure algorithm, learn about the population structure, and basically control for that when we're doing association mapping. And in practice, that kind of concept has been carried through, although the actual methods that people use have used more recently are mainly using different techniques. So, for example, principal components analysis. But the concept of using a statistical method with all of the data to learn about population structure up front has borne through. The second kind of thing that we've used it for, we and many others have used it for, is just kind of understanding how individuals in a sample are related to one another. And so maybe this is getting ahead of ourselves, but there's the paper that Noah Rosenberg and I published around 2002 or 2003 that looked at global population structure and showed that there's relatively strong structuring and that it follows continental groups fairly clearly. And then of course, these kinds of methods of given rise to the like sort of ancestry estimation and direct-to-consumer testing. So if you think about like something like the 23andMe ancestry reports or Ancestry.com ancestry reports, they're kind of using the way of describing ancestry that we had in that original model, though the methods now are a little bit different.
How does the structure algorithm use genotype information to group samples?
One reason why we wanted to do that at that time was that it was unknown what the structuring of genetic variation would be. So we talked about this explicitly in the paper, that you could, up until then, what people did was they would predefine groups and then test for a low frequency differences between those predefined groups, but nobody knew whether those were the right groups or not. And so the point of structure was that you didn't need to use those predefined labels. In retrospect, it turned out actually that often predefined labels do map onto genetic structure quite well, but that it wasn't a foregone conclusion prior to doing that. And the main difference between something like structure and what the DTC companies do now is that now they actually do start from the labels, because it's known that that those map to structure pretty well most of the time. And then the last area where the structure method has been used a lot is in conservation genetics. So it's had a huge impact there, because one of the main goals in conservation genetics is to identify distinct populations that need to be that need their own protection. And so under legislation like the Endangered Species Act, the government is required to protect genetically distinct groups. And so methods like structure have had a huge impact there.
How does the structure algorithm handle genome scale data?
So one big difference is that the original programming that I did doesn't really handle genome scale data very well. So the most widely used method now for genome scale data is called admixture, which comes from a UCLA group, and that has a lot of the elements of the original structure model, but it's got a lot of computational speed ups that make it work well for that. And then there are a number of other flavors of models that have jumped off from the original structure model that have better performance in certain kinds of settings. So, for example, fine structure from Daniel Falouche and others uses a model of haplotype copying followed by clustering, and so in some ways the goals are similar to the original structure model, but it can actually get much better accuracy in settings of very fine population structure. So they showed, for example, that in when applying this to the British Isles, that they can get really exquisite levels of structure. So, for example, individuals from Cornwall cluster separately from individuals from Devon, even though those are adjacent counties.
From your perspective, what were some of the key findings of the HapMap project?
HapMap was incredibly important from the point of view of giving us the first genome scale look at variation. So prior to that, everything was pick a few regions of interest, put some SNPs in there, or do some sequencing, and we just had a very, very limited view of what genetic variation looked like, both within populations, between populations. And HapMap was the first time that we actually had a genome scale view of variation. And one thing that my lab was particularly interested in, as well as a number of other groups, was identifying selection signals. And so for the first time we could ask what are the biggest signals of selection anywhere in the genome? And so it was just really a major change to be able to have that scale of data. It gives you a fundamentally different feel to know, let's say, by some measures, like the lactase signal for selection is the biggest signal in the genome. People believed that lactase was under selection before HapMap came along, but we didn't know whether that was going to be one signal alongside 100 other signals that would be of similar strength. And and actually lactase really stands out when you have the whole genome as being an interesting signal. At least in Europeans it stands out. The other main contribution of HapMap, I think, was in terms of driving the technology. So it really supported SNP genotyping, and I think that as an outsider, my sense is that the investment that was made in genotyping technology to drive HapMap was really a step up towards being able to do genome-wide genotyping for GWAS. And of course, GWAS, the first GWAS studies came out a couple of years behind HapMap.
What were some of the key accomplishments of the 1,000 Genomes project?
So by the time the 1,000 Genomes started, we were doing quite a lot of work on statistical methods for understanding genome-wide association study data, and so prior to 1,000 Genomes, all of the work was assuming that you've got a framework of SNPs, but a relatively coarse framework of SNPs, so that generally the causal variant would be unobserved. And so we put a lot of work into methods where we would try to infer the presence of something that you couldn't observe directly. And so what 1,000 Genomes did was it really transformed the way we could think about the data. So when you had 1,000 Genomes data, then you suddenly knew that the causal variant, so long as it's reasonably frequent in the population, is actually in your data set. And so now the way that you think about the data is greatly simplified. And so, that really transformed the way that we thought about all these kinds of problems. The other kind of advance, I think, that was really important coming from 1,000 Genomes was technology. So, obviously they pushed genome-wide sequencing forward a lot. But the other thing I would mention particularly is imputation. So at the start of the 1,000 Genomes project, there was quite a big debate about whether they should do a small number of individuals at 20 or 30x coverage so that they could get good genotype calls from each individual, or should they do a large number of individuals at something like 4x coverage. And they ended up doing a mixture of both, but it's tilted towards doing a large number of individuals at relatively low coverage, combined with imputation methods. And so, one of the things that they showed was that kind of approach can work, and so that's influenced a lot of subsequent work, that kind of technical innovation.
What has been the significance of recent studies on structural variation?
So I think that over time, people's views on the contribution of structural variation and rare variation to disease has sort of ebbed and flowed over time. So we were actually involved in one of the early studies showing that deletions in the genome are relatively common. And so around 2007 or 2008, when it was becoming clear that the genome-wide significant hits from GWAS studies didn't explain a lot of the heritability, a lot of people thought that actually maybe structural variants were going to be a big part of the picture. And for complex traits, I think now the answer is that they're a relatively small part of the picture for contributing to heritability to common complex traits. However, of course, they are important for they're important contributors as major effect loci to particularly to early onset diseases in some cases. So structural variation is certainly an important piece of the puzzle, but I think it's fair to say that in terms of the percent of variation that it explains, it's a relatively modest fraction of everything. And actually, I think we could say make similar comments for rare variants.
Can you describe the concept of missing heritability?
So by around 2006, 2007, 2008, there was the first set of genome-wide association studies were rolling out, and these studies were typically finding anywhere from, let's say, 5 genome-wide significant hits to 50, depending on the trait. And so one of the calculations that various people did was to take the estimated effect sizes of those lead variants, and from that to try to estimate how much of the the expected heritability could be explained by those. And here the heritability refers to basically the similarity between relatives that's estimated to be due to genetic effects. And so the general observation, which was found across a whole lot of different traits, was that that first batch of genome-wide significant hits was only explaining anywhere from, let's say, 1 to 10% of the expected genetic contribution to these traits. And, these were based on studies with thousands of cases and controls. And so this raised the question of, if you can only explain, let's say, 10% of the variance of the expected genetic variance in height based on the first 30 loci, then, where's all the rest of that familial similarity coming from? And so I think that, the term missing heritability, I'm fine with that term. I think it's a nice evocative term that, kind of summarizes the question. And so at that point, there were a whole bunch of different possible hypotheses. And there's a paper from Terry Manolio and colleagues in Nature, as you know, that basically lists these different hypotheses. And so one hypothesis was that there's a large number of additional common variants that are not genome-wide significant, and these add together to explain the rest of the variants. So it was clear at that point that there would be additional alleles that were not genome-wide significant. The main question was whether those would add up to a small amount or the rest. And then there were other ideas, like it could be structural variation that wasn't very well measured, or rare variants, or maybe inherited epigenetic effects, or other kinds of ideas that were being floated at that time. And since then, I think it's fair to say that the consensus that we've landed on is that the majority of the missing heritability is actually due to there being very large numbers of common alleles with small effects. And so Peter Vischer's lab in particular was responsible for the first analyses that showed that that's probably the responsible for the lion's share of the missing heritability.
How did studies of height illustrate the concept of missing heritability?
Yeah, so the first paper is from Peter Vischer's lab on height, and for that they showed that that there's a lot of signal in the rest of the genome that's not genome-wide significant. But if you add all of that together, even though you don't, so it's using these methods where it says that even though you don't know which SNPs are actually responsible, you can estimate how much total signal there is in among the non-significant variants. And if you add all of that together, in some sense, it's kind of adding up to explain most of what's missing. And then I think that one thing that didn't really answer yet was how many of those variants there were. And over the next few years, one thing that started to become clear was that there's actually an enormous number of those alleles that are not genome-wide significant, and that they span across a large fraction of the genome. So Alcus Price, for example, had a paper that showed for schizophrenia that if you chop the genome up into 1 megabase blocks, that in their estimate, anywhere from 70% to 100% of the 1 megabase blocks contain non-zero amounts of heritability. So the heritability is sort of spread across the whole genome. There are no loci in the genome that are contributing a lot of the heritability, and most of the genome is contributing something.
Can you describe the omnigenic model of genetics?
So starting maybe 3 years ago, we were working with a number of different genome-wide association studies data sets, and one thing that was striking to me was by various measures just how such a large fraction of SNPs or genomic windows show signal of association. And as I alluded to a moment before, there are other people have also observed similar sorts of things. So, for example, Alcus Price's work also points to that. And so actually, this kind of started as a thought experiment. what would it mean if our null hypothesis actually is that, maybe instead of there being a relatively small fraction of genes that can have effects on the phenotype you're looking at, what if essentially every gene that's expressed in a relevant tissue can matter? And so that's basically the this omnigenic model that we proposed. So essentially what that hypothesis says is that if you take the set of genes that are expressed in the cell types that are relevant for driving a particular disease, then potentially any of those genes can have non-zero effects. So then we start to think about what kinds of models could fit that. So like if what kind of mechanistic process could give you that sort of result, and so we've done a fair bit of modeling work on this, and so the kind of model that we like is that there's a set of genes that are functionally close to the phenotype that you're interested in. So we refer to these as core genes. And so specifically, we define core genes as being the set of genes where there's kind of a direct causal arrow from the genotype or expression level of that gene that goes to the disease phenotype that's not flowing through gene regulatory networks inside a cell. And then all of the other genes that are expressed in those disease-relevant cells, they don't have direct causal arrows to the disease. We refer to those as peripheral genes. And because of the way we set up that model, that implies that the only way that they can matter is that their effects, for example, their gene expression is perturbing regulatory networks in such a way that it perturbs the behavior of core genes. And so what we believe is that, based on both observation and mathematical modeling, is that although the biggest signals probably often come from core genes, the core genes themselves actually only contribute a small fraction of the heritability. So that it depends a little bit on the structure of the network, but you can actually for quite reasonable models, it turns out that you can have a very small fraction of the total heritability is coming from these core genes, even though in some sense biologically they're the most important. And so, this kind of connects to the missing heritability argument, because it's, in the sort of preconceived notion that we had before GWAS studies was that there'd be, some limited number of genes that are relatively close to the phenotype, and so when you do mapping, that the hits should concentrate in those or in a subset of those. But instead, what we think is happening is that there's actually, a huge number of genes that are contributing, and the genes that are biologically proximate to the phenotype only contribute a relatively small amount to heritability.
Does the omnigenic model work for simple and complex disorders?
So we think that this kind of pattern is quite wide-ranging. So we have some unpublished work where we've been looking at small molecules or other biomarkers from the UK Biobank. And so these are often things like urate or testosterone, as well as lipid levels. So these are kind of intermediate phenotypes where a lot of the biology is known for these. So we know often what are the metabolic pathways that produce these molecules, or what are the key transporters. And so that seems like a setting where, perhaps one would expect that actually those genes that we know should play a role, maybe you think those should explain a lot of the heritability. Well, actually what happens is very interesting. So we find that the biggest hits are very heavily concentrated on the genes that matter, but they still only explain a modest fraction of the heritability. There's a genome-wide background that's again, a highly polygenic signal. It's probably not as polygenic as it would be for something like height or schizophrenia, but the qualitative pattern is somewhat similar. So you get a sharper signal on the genes that we know should matter, but still on this polygenic background. And I think what probably happens with more complex traits is that they're composed of many different independent pieces that are all kind of flowing through towards the same phenotype, and each of those independent pieces has this kind of structure. And then when you when you map the end phenotype like height, then you're basically superimposing this large number of intermediate kinds of signals, and and that quickly begins to implicate a large fraction of the genome. We actually think that it's really important to think about conceptual models for for why so much of the genome is involved in any given complex trait. So obviously there's been a lot of focus on trying to understand what are the top hits for typical traits, and that's rightly so. But I think that it is a surprising finding of the last 15 years that probably tens of thousands of variants contribute to a typical complex trait spread thinly across and widely across the genome. And so what kinds of conceptual models would produce a signal like that? And so our conceptual framework is one attempt to explain that. And I think that's something that can we can debate the merits of that model, but it is important for the field to try to come up with conceptual frameworks that can explain that, whether or not our particular model winds up being exactly correct or not. I mean, the history of human genetics was people systematically, overestimating their power. So, as in the 1990s, people did association studies with a few hundred individuals, and and almost nothing came out of any of that work. And then, by the mid-2000s, studies had gotten up to a few thousand cases and controls, and that was enough to, usually to detect a handful of the biggest hits. And now that the biggest studies are, meta-analyses of on the order of 1 million individuals or more. We're getting many hundreds of hits, but it's still, detecting confidently only a small fraction of the true set of loci.
What are your thoughts on the development of polygenic risk scores?
I mean, it's still a little bit complicated, and nobody's fully dissected exactly what's going on, but we have some reasonable idea about that. But if it's the case that actually the accuracy of PRS scores within a population depend very strongly on all of these environmental covariates, then that does suggest that the implementation of polygenic scores is going to be complicated. Like we need to think harder in the long run about what exactly environment means, and how the phenotype is being produced by some combination of environmental factors and genetic factors. And I am optimistic actually that polygenic scores will become useful in the clinic, but I think that there are a lot of issues that need to be worked out around this, and some of those issues relate to porting across populations, but some of them relate to things that we don't really understand at all. And actually, I mean, it's worth noting, of course, that the results showing low portability across populations, while in some part those are surely due to genetic differences between the populations, there's very often major environmental differences. Nobody really has a good measurement of, the relative contribution of allele frequency differences and environmental differences. And I'm sure that environmental differences are important.
What are the important contributions of the Human Genome Project?
I think it's just stunning the rate of progress that we've seen in this science during the last 20 years. So I like to tell my students that, during my postdoc, I never encountered a data set that was too big for me to open it up on the screen and scroll top to bottom in the data set. And, that's just unimaginable to them today. And, and that's in part a technology revolution that's allowed us to, measure, measure sequence data, genotype data unfathomable, scales that were unfathomable 20 years ago. But at the same time, that has also completely transformed the way that we understand genetics. And, I think that these questions that we were talking about around polygenicity or the missing heritability or the oligogenic model, I think that those are examples of something where, in the 1990s, before we really had data on this, people kind of imagined that, maybe complex traits would be driven by half a dozen loci. And, the fact that they're probably driven by tens of thousands of loci is just, completely, eye-opening and baffling if you look at that from a 1990s viewpoint. And that's just one example of 100 different ways that the Human Genome Project and everything that's followed after it are it's completely transformed our view of biology. There's this paper from another paper from Neil Risch that I like to cite, which is from 1999, where they had a SIB-pair study of autism, and they, initially they thought they would be powered to find loci that affected autism. In fact, they didn't find anything. But the beauty of the study is that they didn't stop there. Neil had these very careful calculations to look at how many loci there would have to be in order for them to have done a study of this size and in fact not find anything. And, and he estimates it's about 10 or more. And, and that was actually a big deal. I remember reading that paper for the first time in 1999 and thinking, wow, that's really surprising. There's a lot of loci, and, it just seems ridiculously low now. And to your point about technology, I think, I've drawn one example that, relates closely to what we've been working on, but, of course you can tell a similar story about almost any aspect of biology that relates to genetics and genomics, where, the new findings that have been enabled by technology that, came from the genome or the $1,000 genome sequencing and all of the related assays, all of those areas have been completely transformed by that. Yeah, and you can take the, pocket sequencers out into the field and, in remote locations and sequence on site. You could, potentially do, clinical sequencing of individuals in remote locations. It's very exciting.
What areas of research are you excited to pursue in the future?
I continue to be really interested in this, this sort of omnigenic question, why does so much of the genome matter? And so, we have, as I said, we have this hypothesis that, most of the heritability is flowing through gene regulatory networks. But, these are largely unknown or unmeasured. Until now, we just really haven't had tools for measuring those. And so one kind of thing that I'm very interested in is, is using new technology. So, for example, CRISPR perturbations followed by some kind of measurement of gene expression to, start mapping these out. And I think there's a whole bunch of different technologies that are in this kind of space that are starting to allow us to get a picture of the sort of the regulatory structure of cells, and how are different gene products interacting with each other to produce cellular phenotypes and ultimately organismal phenotypes. And there's really an enormous black box from genetic variant to end phenotype. And so that's one of the things I'm really excited in for the next 5+ years, is to start seeing how that piece begins to get filled in.